Now let us follow aerobic respiration, okay, and the equation in brief. What happens during aerobic respiration? What are the steps involved? Aerobic respiration is the respiration that occur in presence of oxygen. Of course, I can say by utilizing oxygen. Oxygen play an important role and direct role in aerobic respiration, and it is utilized. And as we follow the equation, if we apply aerobic respiration process for one glucose molecule, one glucose undergo aerobic breakdown by utilizing oxygen. Of course, 6 oxygen molecules utilized for one glucose and undergo complete oxidation, that is by 6 oxidation steps, okay, and finally broken down into 6 molecules of carbon dioxide while evolving 6 water molecules, and the total energy present in the glucose molecule get released stepwise, and that is equal to 686 kilocalories of energy, okay. And now let us follow the steps involved in aerobic respiration. And of course, the first step in any kind of respiration is glycolysis. So aerobic respiration, of course, the products of glycolysis only continue aerobic respiration. So we may consider the first step as glycolysis that occur in cytosol. There one glucose molecule already we followed how it undergo stepwise reactions and finally split into 2 molecules of pyruvic acid, and the 2 molecules added to cytosol at the end of glycolysis, and they involved in aerobic respiration in presence of oxygen and of course by utilizing oxygen. So the fate of pyruvic acid as we follow in anaerobic process and aerobic process. And pyruvic acid in anaerobic organisms in the absence of oxygen undergo fermentation, whereas the fate of pyruvic acid in aerobic organisms in presence of oxygen based on availability of oxygen, and they continue aerobic respiration, that is with oxidative decarboxylation of pyruvic acid, in other words called link reaction, and TCA cycle, that is Krebs cycle reactions, and then the glucose molecule fully broken down into carbon dioxide and water. And here link reaction and Krebs cycle that occur in the matrix of mitochondria. Link reaction and TCA cycle, that is Krebs cycle, these 2 reactions occur in matrix of mitochondria. So mitochondria is involved in aerobic respiration, whereas formation of pyruvic acid from glucose molecule, that is glycolytic pathway, anywhere occur in cytosol, okay. So, an aerobic respiration process include glycolysis, link reaction and Krebs cycle, that is TCA cycle. Glycolysis occur in cytosol, whereas link reaction and TCA cycle, Krebs cycle occur in the matrix of mitochondria. But by the end of TCA cycle or Krebs cycle, what we observed is and glucose completely broken down into carbon dioxide and water, and energy get liberated in stepwise manner, but majority mean the major part of energy liberated, that is packed in the form of NADH2 and FADH2. Only few molecules of ATP are directly synthesized. So cell is with NADH2 and FADH2 molecules now, but may not be utilized for cellular activities. So the energy packed in the form of NADH2 and FADH2 also should be converted and again should be packed in the form of ATP. For that, the last step of aerobic respiration takes place, that is electron transport mechanism and oxidative phosphorylation. Oxidation of these reduced coenzymes and liberate electrons and protons which are involved in the synthesis of ATP. Based on Peter Mischel chemiosmosis, already we know how electron transport cause ATP synthesis. So like that we followed aerobic respiration, a brief explanation, and how a glucose molecule undergo complete breakdown or complete oxidation with 6 oxidation steps in presence and by utilizing oxygen and liberate the total amount of energy. But of course, the steps involved is glycolysis, link reaction, TCA cycle, that is Krebs cycle, and electron transport system and oxidative phosphorylation. As we follow the venue of aerobic respiration, may formation of pyruvic acid occur in the cytosol, during glycolysis, but the remaining steps occur in mitochondria, and mitochondria play an important role in aerobic respiration. Students, now I ask a question, and maybe anaerobic organisms and aerobic organisms, as we compare, anaerobic organisms do not require mitochondria. Yeast, of course, a eukaryote even had mitochondria, but mitochondria is not involved in anaerobic respiration, we followed. In the same way, and is there any such organisms that lack mitochondria perform aerobic respiration, do you know? Yes, of course. You know, prokaryotes, totally devoid of mitochondria and no membrane bound cell organelle they had. So, and aerobic prokaryotes, as we recollect, let us take the example, certain bacteria which are aerobic. Yes, there are so many aerobic bacteria, aerobic prokaryotes. Let us consider cyanobacteria which are aerobic, and how that aerobic prokaryotes that lack mitochondria perform aerobic respiration without involvement of mitochondria. Is it happen? Yes, of course. And there, and in the absence of mitochondria in prokaryotes, aerobic respiratory mechanism is performed by cell membrane, and previously may you followed certain bacteria develop infoldings of cell membrane called mesosomes, and mesosomes are involved in aerobic respiration, and of course cell membrane is there to perform aerobic respiratory mechanism in prokaryotes that lack mitochondria. So, and there are certain aerobic organisms that lack mitochondria but perform aerobic respiration. Example, that aerobic prokaryotes like cyanobacteria and certain bacteria, and perform that by cell membrane, that is plasma membrane. Okay, that compensate the absence of mitochondria. Students, we followed the theme of aerobic respiration and the 4 different steps in an aerobic respiration. Start with glycolysis, then oxidative decarboxylation of pyruvic acid, that is link reaction, and formation of the link substance acetyl coenzyme A, then Krebs cycle reactions, okay, and so that complete breakdown of glucose into carbon dioxide and water. Later, the major part of energy which is packed in the form of ATP and NADPH2 and FADH2, but most of the energy is packed in the form of NADH2 and FADH2, and that is converted into ATP again. In the last step of aerobic respiration, we followed electron transport and oxidative phosphorylation. Now let us follow the elaborate mechanism of aerobic respiration, and of course we followed glycolysis already in detail, and one glucose molecule split into 2 molecules of pyruvic acid, and the 2 molecules of pyruvic acid added to cytosol, and there in the presence of oxygen, pyruvic acid molecules added to mitochondria, and with pyruvate translocator we followed, okay. And then what happened to that pyruvic acid there in mitochondrial matrix? Now let us discuss. So now we are discussing the phase of aerobic respiration that occur in the matrix of mitochondria, and that include oxidative decarboxylation of pyruvic acid molecules into acetyl coenzyme A, and the complete breakdown of acetyl coenzyme A into carbon dioxide and water. So the phase of aerobic respiration that occur in mitochondrial matrix we can separate into 2 side headings, and that is oxidative decarboxylation, in other words called link reaction, and the next part and the major part that occur in matrix of mitochondria is Krebs cycle, in other words called TCA cycle, tricarboxylic acid cycle. Okay. Now let us continue that oxidative decarboxylation of pyruvic acid occur in mitochondrial matrix. Okay. Now here for 1 glucose we consider 2 molecules of pyruvic acid, and the 2 molecules of pyruvic acid which are added to mitochondrial matrix undergo oxidation, decarboxylation as well as condensation. Okay. And here to perform the reactions oxidation, decarboxylation and condensation, and a complex of or a cluster of 3 enzymes and that enzyme complex called pyruvate dehydrogenase complex involved and catalyze this particular reaction, that is conversion of pyruvic acid into acetyl coenzyme A. Now here we can follow that one glucose molecule and represent 2 pyruvic acid. In oxidative decarboxylation we follow 2 pyruvic acid because we are counting for 1 glucose, and the 2 pyruvic acid molecules, which are 3 carbon compounds, the substrate for oxidative decarboxylation undergo decarboxylation evolving 2 CO2 molecules catalyzed by the enzyme pyruvate decarboxylase, and then oxidation of pyruvic acid results the release of electrons and protons captured by the coenzyme NAD plus. Here for 2 molecules of pyruvic acid oxidation, and we followed the formation of 2 molecules of NADH2. So the major part, the part of energy liberated by the oxidation of pyruvic acid here, packed in the form of 2 molecules of NADH2. For 2 molecules of pyruvic acid, 2 molecules of NADH2 we followed. Then here finally to the product, coenzyme is condensed. So finally the product formed at the end of oxidative decarboxylation as well as condensation, and that is acetyl coenzyme A, which is a 2 carbon compound. Here we follow 2 molecules of pyruvic acid undergo decarboxylation, oxidation and condensation and form 2 molecules of acetyl coenzyme A. And this particular complex reaction is catalyzed by an enzyme complex pyruvate dehydrogenase complex, which is a cluster of 3 enzymes, pyruvate decarboxylase and pyruvate dehydrogenase and transacetylase, which is involved in condensation of the product. Okay? And along with that, the metal ion cofactors magnesium and as well as the proteins and the other cofactors TPP etc. are also involved. And so here we followed oxidative decarboxylation of 2 molecules of pyruvic acid into 2 molecules of acetyl coenzyme A, and during this oxidation step 2 molecules of NADH2 as energetics formed and added to mitochondrial matrix. Now this acetyl coenzyme A is the substrate that continue Krebs cycle reactions, that is TCA cycle. So, and the substrate that start Krebs cycle reactions is acetyl coenzyme A, and which connect the glycolysis to Krebs cycle. So, acetyl coenzyme A is considered as link substance. Okay. So, and the link substance acetyl coenzyme A, which is the link between glycolysis and Krebs cycle, is formed by oxidative decarboxylation of pyruvic acid. So, this particular reaction is named as link reaction. Okay students, now let us continue Krebs cycle or TCA cycle in detail. Students, now let us follow the detailed mechanism of tricarboxylic acid cycle, that is TCA cycle, and in other words also can be referred as Krebs cycle, because this particular biochemical pathway was traced out by Sir Hans Krebs. So, it is also referred as Krebs cycle, and that occur in mitochondrial matrix. Okay. And the substance that start Krebs cycle reactions is acetyl coenzyme A, which is the link substance between glycolysis and Krebs cycle we followed. Of course, acetyl coenzyme A formation occur in the matrix itself, that is by the oxidative decarboxylation of pyruvic acid, already we followed. Now, for 1 glucose, we consider 2 molecules of acetyl coenzyme A. Okay. And so, here we follow 2 turns of Krebs cycle. Here in the scheme, we follow 2 turns of Krebs cycle because we consider 2 molecules of acetyl coenzyme A here, and that is for 1 glucose. Okay. Now, let us follow what happens during Krebs cycle reactions, right. As already we said, it is traced out by Sir Hans Krebs. So, the name Krebs cycle, and the other name for Krebs cycle is tricarboxylic acid cycle we said, that is TCA cycle, because as we verified, the acids that are formed in Krebs cycle reaction, the first one, that is citric acid, and then oxalosuccinic acid, and these are 6 carbon organic acids with 3 carboxyl groups. So, they are tricarboxylic acids. So, the first products that are formed during Krebs cycle reaction are tricarboxylic acids. So, the pathway is also named as tricarboxylic acid cycle, TCA cycle. And what are the tricarboxylic acids here represented? Citric acid and oxalosuccinic acid. Okay. Now, coming to the biochemical reactions and in sequence. The first one, the substance that start Krebs cycle reaction, and that is molecules of acetyl coenzyme A. Here we had taken 2 molecules, and that is for 1 glucose and 2 turns of Krebs cycle here. The 2 molecules of acetyl coenzyme A accepted by the acceptor, that is oxaloacetic acid, which is a 4 carbon compound. So, 2 molecules of OAA and 2 molecules of acetyl coenzyme A condensed, and the reaction is condensation, and the product of condensation is 2 molecules of citric acid, which is a 6 carbon organic acid, and that is TCA also, tricarboxylic acid. Now, 2 molecules of citric acid undergo oxidation. This is the first oxidation in Krebs cycle reactions, oxidation 1, but overall in aerobic respiration this is third oxidation. Previously already 2 oxidations completed, 1 oxidation during glycolysis already we followed, 1 oxidation that occurred during link reaction, that is conversion of pyruvic acid to acetyl coenzyme A. So, in Krebs cycle this is first oxidation, in the matrix it is second oxidation, but overall in aerobic respiration this is third oxidation, and the product of this oxidation is 2 molecules of oxalosuccinic acid, which is again a TCA, tricarboxylic acid formed, but the electrons, protons liberated during the oxidation process, and they are captured by the coenzyme NAD plus and NADH2 formation observed here. And for 2 molecules, we had taken 2 molecules of NADH2 here. So, the energetics for this first oxidation step in Krebs cycle is 2 molecules of NADH2. Then this 2 molecules of oxalosuccinic acid undergo decarboxylation, the next reaction, and the product of decarboxylation alpha ketoglutaric acid, which is a 5 carbon compound. Okay. So, 2 molecules of alpha ketoglutaric acid are formed, and due to decarboxylation of 2 molecules of oxalosuccinic acid. Now, as we verify, and here in Krebs cycle, the substrate itself start reaction is 2 carbon compound, and the first products formed are 6 carbon compounds, and alpha ketoglutaric acid is the 5 carbon compound as a result of decarboxylation we followed. As we verified the other, and the remaining are 4 carbons we observed up to here. So, we consider this alpha ketoglutaric acid as the only 5 carbon compound involved as intermediate in Krebs cycle reactions, and it has a big story. And alpha ketoglutaric acid, it may deviate from Krebs cycle pathway and may involved in the synthesis of amino acids like glutamic acid. Okay. So, it also support amino acid synthesis, that is anabolic process, that we will discuss in the later topic, amphibolic pathway. So, now and 2 molecules of alpha ketoglutaric acid now undergo oxidative decarboxylation as well as condensation, a complex reaction, that is oxidation, decarboxylation and condensation. So, as a result, here decarboxylation we observed, and oxidation of that alpha ketoglutaric acid release energy in the form of electrons and protons, and they are received by NADP plus, and formation of 2 molecules of NADH2 here we observe. And these are the energetics for second oxidation reaction in Krebs cycle, that is oxidative decarboxylation of alpha ketoglutaric acid. And the product formed is 2 molecules of succinyl coenzyme A, because here condensation also occurred. Now, the 2 molecules of succinyl coenzyme A undergo cleavage reaction, and during cleavage reaction certain amount of energy get liberated, and that energy is utilized to couple GDP with inorganic phosphate, and the energy here utilized for the coupling of 2 molecules of GDP, because we consider 2 turns of Krebs cycle. So, 2 GDP with inorganic phosphate form, 2 molecules of GTP. So, here formation of 2 molecules of GTP we observed, that is direct synthesis of GTP molecule as the energy liberated from the substrate directly utilized. Okay. So, this kind of phosphorylation process is called substrate level phosphorylation. Previously we followed in glycolysis. So, SLP also occurred during Krebs cycle. Now we observed, and the result of SLP, 2 molecules of GTP for 2 turns of Krebs cycle we followed. And the product of cleavage reaction and 2 molecules of succinic acid and these 4 carbon organic acid now undergo oxidation, and here particular oxidation is catalyzed by an enzyme called succinic dehydrogenase, and there the energy liberated is packed by FADH2 molecules. So, 2 molecules of FADH2 formation here we observed as succinic acid undergo oxidation and converted into fumaric acid, catalyzed by the enzyme succinic dehydrogenase. Now, the 2 molecules of fumaric acid converted into 2 molecules of malic acid, 4 carbon organic acid, and now the 2 molecules of malic acid undergo the last oxidation step in Krebs cycle reactions, and the energy captured by NAD plus and 2 molecules of NADH2 formation here we observe. And these are the energetics for the last oxidation in Krebs cycle reactions. And FADH2 molecules are the energetics formed during third oxidation of Krebs cycle reactions. And finally, here for 1 glucose, and we followed 2 acetyl coenzyme A involved in Krebs cycle, and this represent 2 turns of Krebs cycle, and for this 2 turns of Krebs cycle, and mean by the time the 2 acetyl coenzyme A molecules completely broken down into carbon dioxide and water, meanwhile, and 6 molecules of NADH2 are formed, 2 molecules of FADH2 are formed, and 2 molecules of GTP by SLP. Total 4 oxidations we observed, 4 steps, and total 8 reactions we may consider. And for that 4 steps of oxidation, during 3 steps of oxidation, NADH2 molecule formation, and in 1 step of oxidation, FADH2 molecule and GTP is formed by SLP reactions. So, these are the energetics that are formed for Krebs cycle reactions. If 1 glucose involved in aerobic respiration in the form of 2 acetyl coenzyme A molecules completely broken down into carbon dioxide and water by the time, and there in Krebs cycle reactions 6 NADH2 and 2 FADH2 and 2 GTP molecules added to mitochondrial matrix.